I'd like to, it's my great honor to introduce Dr. Asia Ryder today as our speaker. She's not really a visitor, visitor to CRINA. She received her bachelor's here at the University of Alberta in infection and immunity. She received it in the honors program. And then she moved to McGill University, where she completed a master's in neuroimmunology, and then moved back here to the University of Alberta, where she completed a PhD in comparative immunology with Dan Beretta.
Yep, that's correct.
And then moved to UC Berkeley to do a postdoctoral fellowship with Ellen Roby using more imaging technology. And I learned just today it was on toxoplasmosis. Then we were very fortunate here at the University of Alberta to recruit her in 2015 to be the flow core manager. And I have to say, she has done an amazing job with the flow cytometry facility, making sure that we have the latest and greatest technology, and also making sure that we know how to use that latest and greatest technology. And as a demonstration of her abilities to lead the flow cytometry facility, in 2017, she was named the ISAC Chaired Resource Lab Emerging Leader from 2017 to 2021. And this is an international award that recognizes leaders in the flow cytometry field. In 2022, she received the ISAC Shared Resources Lab recognition for excellence in core operations. So we have that gold medal of approval as well for the flow facility. She's currently the co-president of the Canadian Cytometry and Microscopy Association and a board member of the Human Cell and Differentiation Molecules Council, which is part of the International Union of Immunological Societies, of which I'm currently a council member. So doing a lot of great work internationally, representing our flow cytometry facility outward, but also taking these latest technologies and bringing them to us as well. So that's a really great thing for our facility. The world of single cell analysis has just changed so much. It is just evolving. It's almost impossible to keep up with. Her talk today will focus on recent technological advances that are available right here in the FOMD core facilities, including high parameter spectral flow cytometry, which we've yet to try, analysis of EVs by flow cytometry, and single cell RNA-seq, and how she's going to talk about how you can implement them in your research program. And the title of her talk is Advances in Single Cell Analysis Technologies. And so over to you, Asia.
Perfect. Well, thank you for the very glowing introduction. Hopefully you can't see me blushing on my camera. So without further ado, let's get started. And so today is going to be probably a little bit different than what you're used to in the CRINA seminars. This will not be a research-focused talk because I am a machine nerd. I love machines, I love how they work, and I love telling people about that. So all of this today, though, will be centered on cytometry, which as its essence is really just the measurement of the physical or chemical characteristics of cells or other biological particles at a single cell level, and looking at technologies by which we can do this. Now, when most people think about standard cytometry techniques, it falls into three major categories. We have flow cytometry, microscopy, and genomic cytometry. Most everyone, I'm going to assume, has probably heard of the first two, maybe not the third. So flow cytometry is looking at a number of parameters within a cell population. It's highly quantitative and you can look at millions of cells really quickly. But its major downfall is you have zero spatial resolution. You know what markers your cells are expressing, but you don't know how that expression relates to each other, where that expression is occurring. Microscopy, on the other hand, gives you that spatial resolution. You can see your cells, you can see what they're expressing, However, you tend to be more limited in how many parameters you can measure, as well as how many cells you can analyze. My foray into the world of science started in a dark microscopy closet counting cells for 8 hours a day. So I know the pains of microscopy, and it's well worth it in the end, but it can be a tough technique to get a lot of cells. And then finally, we have genomic cytometry, which is looking at essentially super high dimensional single cell profiling, generally of the genome, so transcriptome, but also now going into the proteome. So you can look at hundreds to thousands or more genes per cell, and usually around 10,000 plus cells. However, again, like any technique, it has potential issues with doublets that you can't detect easily, or things like gene dropout, which I'll talk about later in the presentation. So because of the focus today and the group that I'm speaking to, and I apologize because I'm doing a number of these this fall, and so if you've heard previous talks, there will be a lot of overlap. But I've tailored this one to two major areas that I thought would be most interesting to people doing cancer research. So the first is looking at highly multiplexed cytometry, where you can really use the power of cytometry to gain resolution. So to look at more things in a single cell without having to split it up into a bunch of different samples. And this is really powerful because you can get more details about what your cells are doing out of a very limited amount of sample. And then the other topic I'll talk about is small particle cytometry. This is everywhere right now, whether it be EVs or viruses. And small particles are really biologically important to study, but can be fairly challenging. So I'll talk about a few technologies we have in the facility to do that. So starting off first with spectral cytometry, because, I mean, why not start with my world of flow cytometry at the top? And taking a step back, looking first at how a conventional flow cytometer works. So for those of you who have done flow cytometry, you'll know that there are specific channels, and that's related to the filters that direct light into that channel. And so on your instrument, your various emission spectra are basically separated out into various channels, and you look at the light as it appears through that filter. And that's how you're detecting your fluorochromes. So one detector is assigned to one fluorophore. When we do this, we're ignoring large parts of the spectra, but also some of the spectra may end up in adjacent channels, and we need to compensate or subtract that out. And so that's really the essence of how our standard instruments are working. We're looking at a small slice of light, ignoring or subtracting all other light that it produces. Spectral cytometry, on the other hand, looks at the entire emission of light from that fluorochrome. So instead of having one bandpass filter, we have a number of contiguous bandpass filters that are always turned on and always collecting all of the light of a fluorophore. And so how this essentially works is you have an array of lasers that, similar to a standard flow cytometer, excite your fluorochromes, and then this light is sent off to all 5 detector arrays, and anywhere that light is detected in a bandpass filter, we get a signal. And so this allows us to develop something like a spectral fingerprint, or a fingerprint for a fluorochrome that we use to identify it. So instead of looking at light from a single detector, we're looking at all the light over a number of different arrays and compiling this into a single fingerprint to identify that fluorochrome. So why is this important? Well, if we go back to standard flow cytometry, when we have one PMT for one color, to add more colors, we need to add more lasers and more PMTs. And as you can imagine, this can get very expensive very quickly. So to put it in perspective, on the largest conventional cytometer, they have 10 lasers, and you can detect about 28 colors. And with a 10 laser instrument, you are looking at a very, very multi-million hefty dollar price tag. In spectral cytometry, again, where we have our detector array that measures an entire spectra, we can do this with a more limited number of lasers. So the highest laser count on any of these instruments is 5. And with those 5 lasers, the current published maximum that's been detected is 40 different colors. However, pretty soon, because there was a poster at Cyto this year showing 50 colors, I'm sure that will be the new benchmark. And so really the difference is how we look at these fluorochromes. So if we zoom in on this picture from the bottom a little bit more, we can see the spectral signatures assigned to each of these fluorochromes. So if we look at the top two, we have this brilliant violet 515 signature here and this FITSI signature here. So just as a standard of reference, this would essentially be the light in this blue box here that we would capture on a conventional cytometer. And because of that, these two fluorochromes could never be paired together because they would look the same to the machine. However, by looking at the entire spectrum of light and looking at the individual fingerprints, we can actually co-stain cells with these two markers and discriminate them and see populations with both, which is really how spectral cytometers get their power. It's by allowing us to combine currently available fluorochromes in ways that we have been limited to previously because of how light is being detected. And you can see that same example here where these two are a little bit more different, but the light again would be in the same filter. We can use them together. These two fluorochromes you can see look almost identical. There's just a very slight shift in their spectra, and even those two can be combined in a single sample. So this really is the power of spectral cytometry. Why do we care about this though? Obviously, yes, getting more fluorophores for certain assays might be really important if you want to look at a lot of markers, but living in a realistic world, most people are looking at a number of markers that can be done just on a standard cytometer. So what would be the benefit of switching to this if you don't want to look at 40 colors? And there's really four main reasons. The first off is having more data points. So instead of looking at a single data point for your fluorophore, you're looking at up to 64 different data points that have you better identifying your fluorochrome signature. Because of this, you're also collecting more photons. So because your photons are collected over the entire spectra, there's generally fewer photon counting errors, and this results in tighter populations. So instead of having a larger, more spread out population, fewer photon counting errors shrinks that in and allows you to better detect dim populations that may have otherwise been masked, or populations that would have otherwise been stuck together. The biggest benefit is also more colors with fewer lasers. This allows the instruments to actually be purchased on standard grants. Our 5 laser instrument is a fraction of the cost of what we would need to get a 10 laser instrument. And then for a number of studies, such as when you're studying tumor cells, you can actually extract autofluorescence. So on the instrument, it can read autofluorescence as a fluorescent signature, and you can use this to analyze populations of cells, which can be a really powerful technology for analysis, but also can allow you to look at cells that may have otherwise been masked by that autofluorescent signature. So just to give you an example of what this can look like, again, this is probably going to look really small and don't get lost in the details, but the point of this is all of these populations were found from a single stained tube. So within that tube, with 40 different antibodies added, they were able to fully phenotype all the major cell populations in peripheral blood, right down to like small sub-activation populations. And this is why this is so powerful, because instead of now having to take your panel and split it up and take that single tube into 8 different tubes to look at all these markers, you can now do it all in one tube, which saves you sample, but also allows you to make really powerful marker combination and different population calculations without having to mathematically figure out how the different tubes relate with each other. So in our instrument, this is done by the Cytec Aurora. It was recently brought in about 6 months ago, and it is a 5 laser spectral cytometer. So when I talk about machines, I like to generally tell people what they are best used for, so people have a better idea of if and how it could work in their research. So obviously I've talked about this a lot. So point number 2, high parameter panels. This is an excellent machine for that. But again, that's a fairly limited number of people reaching those levels. So I also like to have it in experiments where you have weird fluorochromes that don't really work nicely on a standard machine. So this could be things like certain fluorescent proteins or certain antibodies that don't generally detect well on a standard cytometer. The really nice thing about spectral cytometry is if it can be excited, it can be detected. And with the 5 laser combination, I don't know of any current fluorochromes or fluorescent proteins that can't be excited by those. So you can look at literally anything you wanted to here that was fluorescent. It's also really nice when you have cells with high autofluorescence because, again, allowing you to subtract this can reveal populations you don't have otherwise be able to see, or you can actually use it to analyze different populations of cells. All right, so that's the Cytec Aurora. If you're interested, come chat with me. So next up, we're going to talk about genomic cytometry. So if we think about cytometry, again, we've talked a little bit about fluorescent cytometry, where we're using light to measure cells. In genomic cytometry, we're using nucleotide sequences to measure our cells. And in this case, generally when people talk about this, they're talking about the world of single cell sequencing, or in our facility, the 10x Genomics instrument. So on this platform, you have a tube of barcoded gel beads that are all different, or a different mixture of signals on them. And these are then combined in a microfluidics chamber to create little droplets where cells are associated with these beads. And so then when the cells are lysed, the RNA gets the coded signal off of the beads so that you get barcoded cDNA that then you can use to figure out what genes were from which cell after you go back and do your analysis. Now, the one thing to note is this isn't just limited to gene expression. It is by far the most common thing we do on there is this 3' gene expression. And so this kit is stocked in the facility and sold on a per sample basis. However, there are other things that can be read on this machine depending on the kit that you end up buying. So one thing is looking also at cell surface proteins, which is through a technique called CytSeq. I'll talk about this more on the next slide. You can also look at your immune clonotype, so sequencing your B cell receptors and T cell receptors with a specific kit and looking at VDJ recombinations. You can look at antigen specificity, CRISPR perturbations, as well as chromatin accessibility with the ATAC-seq. So there's a lot of different ways that you can approach this just by using slightly different kits and slightly different library preps. So one technique that I find is really cool, because I always like to bring things back to protein and not just RNA, is this technique called CytSeq, or cellular indexing of transcriptomes and epitopes by sequencing. It essentially allows you to identify proteins with an oligotagged antibody and the transcriptome on a single cell level. So in the same single cell, you can look at the entire transcriptome as well as the protein signals from any protein you added an antibody for. Now, the really cool thing about DNA barcoding is you have limitless multiplexing. I haven't seen anybody really test the upper limit of this. I think mostly it's limited by how much money you're willing to spend on it, because it can get very expensive very quickly. But because you're not constrained by available fluorochromes, you can have hundreds of potential combinations in your antibody barcode and different antibodies you can add. There's also no compensation required, so you don't need to worry about spectral overlap. Each antibody has a unique barcode that is identifying it. And it does require some optimization. However, luckily, BioLegend has paired up these antibodies with fluorescent ones, so you can do all your titrations not by sequencing. So it can be a lot less expensive that way. So to give you an idea of what this data looks like, a group down in New York looked at immune phenotypes with both standard RNA sequences as well as with the CytSeq method. And what they found was that the two signals matched up really nicely. So you can see in every figure, the islands match the islands, but you'll see in some of them, like the CD11C, which is blown up over here, you have the blue island is really not well populated. There's a lot of blank space. And this is due to something known as signal dropout. And not something that you can predict that you're going to have. It's essentially just signals that don't pick up nicely with sequencing. However, with protein, you can see we have a really nice signal there. And so that's one of the ways that CytSeq helps is with basically rebuilding populations that you lost with signal dropout. Again, you wouldn't know this ahead of time. This is something you'd have to come back and redo. The thing that I found was really cool, though, was when they looked at the CD8 population, by flow cytometry, they could generally get 4 distinct populations. So low, negative, low, intermediate, and high. And they were able to find those same 4 populations in their site-seq data. So it had a really similar dynamic range of detection, which I found was really cool. So that's a really powerful technique, just much more expensive than flow cytometry, but allows you to look at a lot more combinations and really gain the power of looking at both the protein and the transcriptome in the same cell. So next up in this arena is something known as spatial multi-omics. I will preface this by saying that the machines I'm going to talk about now are not ones we currently have in the facility. However, they are machines that we are very, I guess, rapidly looking to acquire, or they're on our radar, and we're hoping to bring them in soon. And this field really kicked off with imaging mass cytometry. So mass cytometry uses, instead of fluorochromes or barcoded DNA, uses metal isotopes to mark the cells or mark the antibodies. And then this is put through a mass spec machine, and that is how you get the readouts for this. So imaging mass cytometry is done on a tissue slice and allows you to analyze up to 37 different protein markers in the spatial context of a tissue microenvironment. And when this came out, that was pretty groundbreaking, because being able to get that many antibodies onto a single slice of tissue was just revolutionary, and you no longer had to do multiple tissue sections to look at all these different markers. And the part that I just love looking at are the images, because I find them just insane in how detailed they can be when you think that the tissue is on a slide, it's been laser ablated off the slide, and then it's been ionized, and then through some magic wizardry has been rebuilt to recreate these really exquisitely detailed images where you can look at all these different markers. And even though they generally still only show them 3 markers at a time, all these tissue slices were stained with 37 different markers and just shown in different combinations. So it was a really powerful technique that kicked off this desire to look at more things in a tissue. And so from this spawned the world of spatial transcriptomics, or looking at your transcriptome sequencing in a tissue rather than a dissociated cell. And the one that's really caught on the most is this one, using an array of spatially barcoded probes. So essentially you have a slide with an array on it, your tissue slice goes on top of that array, and then the cells that are on each spot, that's where they get ligated. And then you can do next-gen sequencing of the captured probes. And so in our world, this is essentially the 10x Genomics Visium platform, is how this is accomplished. And there is a way to do this with our current infrastructure in the high content analysis core. And so what the readout for this looks like is essentially you have your pre-imaged tissue, so you know the general regions in which you want to look. And then you can go back and overlay the spatial transcriptomics over top and be like, oh, this cancer gene of interest is most expressed in this lower region, which is where I suspect these types of cells reside. So it gives you this really powerful spatial context when you look at your gene expression, because you could have done this on a dissociated tissue and looked at all the single cells, but you would no longer know how those cells related to each other. So this spatial context is really important for that aspect of the biology. And this is a world that right now is growing really quickly, especially in the tumor microenvironment world. And looking up this, or looking at spatial data, has opened up new possibilities for studying specific tissue regions and cell types. It's opened up possibilities for looking at local cellular behavior, as well as interactions between diverse cell types, and has really resulted in this nice workflow where all of these various techniques are interacting in order to bring biology forward. So the instrument we're looking at in this world is the Akoya instrument. And to show you kind of what that data would look like is, here they have 100 plex, so 100 different antibodies were put on this tissue with all these various areas in which they were targeted. And then if you look at the tissue images, you can see an overlay of where these were located. And excitingly is this is looking at protein signals, not just RNA signals. And so you're really diving deeply into that biology. You're able to characterize your cells on a high level and also look at spatially how they are related to each other and how that spatial orientation may relate to the biology that you want to study. So a very cool, very powerful technique. Okay, I'm kind of flying through this. I'm sorry if it's really fast. I can't tell if any of you are enjoying it because I can't see anyone. I'm very excited for the days when Zoom talks are over. So switching gears a little bit here, let's get into small particle cytometry. So when we think about cytometry, in conventional flow cytometry, we're generally over in this part of the world where, you know, kind of about 1 micron higher, bacteria might be the smallest thing you look at. And this is really well established in terms of it's been like this for decades. So this new small particle cytometry is focusing more on this world, the small side of things that characteristically people have thought can't be detected on a flow cytometer. And that's generally because the limit of resolution for a flow cytometer, depending on the platform you're looking at, is somewhere in this range. So somewhere between 50-ish and 100 nanometers. Again, this is going to be highly affected by things like refractive index and fluorescence intensity. And so it's not a hard and fast rule, but somewhere in that range is where our flow cytometers really can't see anything anymore. And that has to do because of something known as noise. So a flow cytometer, all the detectors are going to have noise. Some of this will be electronic noise from the detectors themselves. Some of it will be due to small salt particles in your sample, in your sheath. Some of it is dirt, debris, dust, all sorts of things contribute to noise. So if you look at this panel on the far left here, you can see this huge shadow of events. This is going to be a mix of EVs and noise. There's nothing here that's pulling them out of the noise. Whereas the one just beside it is where you have a fluorescent antibody and you can kind of see this population shifting outwards. So when we're doing EV or virus cytometry on a conventional cytometer, that's kind of what we're looking at. There's a huge population of noise with a slight sort of smear coming out of it. Because of this, fluorescent staining is really important. And the brighter you can make this staining, the better, because it will bring you more out of the noise. And in addition to this, controls are extra critical. When you're doing EV work, you don't have the benefit of cells where you can spin them down and wash them. So anything you add to your EVs, like antibodies, introduce more noise into the system, which makes it more challenging to detect your population. So having the right controls is really even more important here to make sure you're actually seeing what you think you're seeing. So in our facility, we have a newly acquired Cytoflex S. This is a small particle dedicated flow cytometer. I have gone through and fully optimized and calibrated this instrument. And so there is a template that is ready to go and no adjustments are needed. So essentially you can walk on, scatter, threshold, and fluorescence have all been optimized. So you just run your sample at those settings. And if you use our template and another software known as FCM Pass, you can standardize your data and also get size information for your particles because of the optimization and calibration. This instrument has 3 scatter channels. So normal forward scatter, side scatter, as well as a violet scatter, which is really important for small particle analysis, and 9 fluorescent channels. Not a lot of channels open here because generally for EV work, you're not doing a huge multicolor panel because there's not enough space on an EV to put that many antibodies. It also has a 96 well plate loader, which can minimize your workflow because you can load the plate up and walk away. So another technology we have, this is not a new one, but another one I'd like to speak about is EV detection by imaging flow. So in the image stream, we essentially have a combination of a fluorescent microscope inside a flow cytometer. So the same sort of laser ray you would generally have activates your cells in the flow cell. And then this is magnified through an objective lens, either 20x, 60x, or 40x. For EV work, it's always 60x. And then this is split off onto 2 different CCD cameras. This is important because this is where the benefit of the image stream comes in. Because it's using a CCD camera instead of a photodiode, there's a difference in how the signal is integrated. And so you get a higher sensitivity, especially in red channels. So essentially, it's easier to detect dim events on the image stream. And as well, the CCD cameras have less noise than a photodiode. So having a signal that is slightly brighter because of increased sensitivity and less noise makes it much easier to find small things. So it's a really nice instrument for EV work. So this is a paper that was published in 2019 comparing the Cytoflex to the image stream, so the 2 instruments we have, and looking at the exact same samples collected on each instrument. So the first thing I want you to look at is on the left, we have the Cytoflex. And again, you see in the circle here, the giant cloud. To the right, we have the image stream. And this is the background noise. So you can immediately see compared between the 2, there's far less noise coming through on the image stream than on the Cytoflex. Now, this giant cloud up in the top of the image stream is not actually noise. Those are the speed beads. They're a calibration particle that's always running in the instrument to keep the camera in focus. So this can easily be removed from your analysis. And so because of the less noise, you can see in these 2 panels, the GFP signal here is really attached to the noise, whereas here it's a much more distinct population. You can see this even more clearly in the next plot with the arrow, where again, the GFP population on the Cytoflex is really jammed up into the noise, whereas on the image stream, you have a really discrete difference between the background and the signal, allowing you to better gate this. And one thing that's also important to note is on the image stream, actually far more events were detected because we're able to see these dimmer events due to the lack of the noise and because it's sensitive enough that they can actually be detected. So those are the 2 big benefits of the image stream. Higher sensitivity, less noise, makes small things easier to find. Now, one thing people don't often think about when they want to analyze small things on a flow cytometer is the fluidics. And the fluidics on a flow cytometer has been designed so that a cell can go through approximately 1 cell at a time. And it's really great at doing that. However, cells compared to EVs are quite large. And so when we start putting EVs or viruses or other small particles through a flow cytometer, we get something that looks more like this or like this. So very rarely are we ever detecting 1 event at a time. In cases like this of extreme coincidence, also known as swarm, your results are highly inaccurate and not really usable. This right here is kind of your best case scenario where your laser is only getting a small handful of EVs at a time. So on conventional flow, this can be somewhat difficult to measure. It involves doing a number of dilutions and basically looking for the point at which you have a linear curve between the number of objects you're seeing and your dilution factor, or your MFI is not changing. So if you have like this, a lot of events here will have a higher MFI than something like this. So when you're in a high level of swarm, your dilution factor will bring your MFI down until it flattens out. So that's when you hit sort of this range. So on a standard flow cytometer, that's more or less all we can rely on to make sure we're only looking at 1 particle at a time, which, as I said, is very rare. On the image stream, we actually can use these images to our benefit in this case. Because we have images, we can see how many dots are in each image. We can start making masks to find these dots, and we can start gating based on the number of dots in an image. So you can see in this same sample where, again, this was actually part of this figure. So where we weren't seeing coincidence based on standard metrics, you can see there's a lot of events or a lot of images captured that have multiple events in them. So we did, in fact, have coincidence occurring. However, using our masking ability, we can gate on the cells or the images that only have 1 event and only analyze those. And so this gives added power to those analyses because you know that you're never working in coincidence and definitely never working in swarm. And so an added extra benefit of having an image here. And another final cool thing about the image stream, I talk about this machine a lot because I am a bad flow core parent and I pick favorites, and this machine is my favorite, is it allows you to have an increased perspective on your biology because you can look at both cells and EVs in the same sample run with the same settings. So as you can see here, just based on different gating strategies or gating different parts of the populations, up in this top corner, we have our cells. Then we have probably apoptotic bodies, and then smaller and smaller microparticles that were detected. And these are in the different gates to the right hand here. Now on a conventional flow cytometer, this is pretty much impossible because in order to see your small particles out of your noise, you need to run your scatter on a log scale. This automatically pushes your cells off scale and are undetectable. If you want to run cells or see cells with your scatter, you're running on a linear scale. This pushes your small particle into the noise and they are no longer resolvable. So the image stream is, as far as I'm aware, the only technology that allows you to analyze right from cells down to the smallest visible EVs in the same sample. So this is the image stream. It is a 12-channel instrument with 9 fluorescent channels. As I mentioned, 20, 40, or 60 times magnification. I've talked a lot about how it is really great for small particle analysis. However, it has a lot of other features in which it is really stellar. Some examples are phagocytosis, nuclear translocation, cell-to-cell interaction. It's really good also at cell cycle analysis, or really any assay where having the added resolution of an image is beneficial, which is a lot of assays out there. So also recently in the facility, we've brought in the ExoView R200, which is an automated exosome and virus imager. It runs on a series of microarray chips that are purchased from the company there's various kits that they have available for purchase. And they can also make bespoke kits for you if you contact them. They can counterstain up to 4 fluorescent colors. And this instrument allows you to compare exosome size. It can do counts, co-localization, protein expression, and exosome cargo content, kind of. So how this technology works is you have a microarray chip that has a distinct antibody array that's already printed onto it. To this, you add your EVs. They're captured by these various antibodies, depending on what they express. And then you add your fluorescent detection antibodies. It's really just like a fancy fluorescent ELISpot. And so based on this, you get this beautiful spotted picture in which you can look at your EV phenotype based on the fluorescence. And then you can also look at co-localization and measure the diameter based on these images. Now this technique, its, I guess, claim to fame is the ability to permeabilize and look inside an EV. So you can imagine a standard EV floating around. If you try to permeabilize it like you would a cell, it basically just explodes. This is also generally one of the standards we do in flow cytometry is add a detergent. If all your signal disappears, it was an EV because all your EVs have been destroyed. And so finding a way to look inside an EV has been something that the field has been looking for a long time. And this is the closest we've gotten so far. So your EVs are captured based on transmembrane protein expression. They're then fixed onto the chip, permeabilized, and this allows you to stain the internal EV membrane. So it's not going to allow you to look at things like the RNA cargo, or if there's any other things floating around inside of the EV. It allows you just to look at the things on the inside EV envelope, which is also very cool. Okay, so that's our newest instrument. Again, if you're interested in this one, there are a number of different kits available. I can put you in contact with their sales rep and they can help you build or purchase whichever kit you like. So that is a lot of information on some of the cool new tools we've acquired. There's a few other machines I wanted to talk about just because they're really nice complementary techniques, even if they're not the most cool cutting edge machines on the market. So the first one that we've brought in is the AutoMax Pro. This is our newest instrument to the flow core. It's been in place for about a month. And what it does is essentially automatically run your Miltenny microbead separations. So the instrument will add the beads, perform the separation on your cells in a very automated manner. It's all time controlled and pipette controlled, and essentially you load everything up, program it, and walk away. You can also run any Miltenny product on it manually, where essentially you add the beads and the instrument does the separation for you. And it is more cost effective than running individual separation columns. So starts at $18 separation with discounts available after 10 or 20 in a month. So if anybody is doing Miltenny separations in their lab, feel free to come chat with me. We also have a lunch and learn coming up on November 2nd, where they will talk more about this technology. Next up, we have our new user-operated sorter, which is the Sony MA900. This is a fully automated cell sorter, and training is available to any user who is interested. It's also a BSL-2 instrument, so you can sort primary human samples or infected samples on this instrument. It is 4 lasers and 12 fluorescent channels, can sort 4-way, so 4 different populations simultaneously into 4 different tubes. Or if you want to do 2-way sort into larger tubes, you can bulk sort onto plates or single cell sort into 96 or 384 well plates. It has different sort modes available for different types of applications, ranging from ultra yield, where basically you're trying to capture as many cells as possible, to ultra purity, which is what we use for genomic applications to get the purest population possible. As well as multiple nozzles available to sort anything from lymphocytes all the way up to 130 would be kind of like the size of an amoeba. And then it has something called targeted sort mode, which is specifically designed for large or atypically shaped cells. So if you want to sort tumor cells, this would allow you to do that more seamlessly because it will change the shape of the droplets so they can be better captured and sorted. So really cool machine, really friendly to use, and really well loved by the users so far. In the high content analysis core, we've also recently brought in the Seahorse Metabolic Analyzer. This instrument measures the oxygen consumption rate and extracellular acidification rate, so general metabolic outputs to measure respiration and glycolysis. We have the 96 channel format, and though the plate can be used once and 96 wells are analyzed, we do stock the plates in the core, so you can purchase them directly from us. And there's a very wide range of applications depending on the kit that you choose and purchase. In the high content analysis core, we also have a bank of short hairpin RNA plasmids that you can use for any sort of gene knockout studies. So there's both human and mouse available. If you're interested, contact myself or Mike and we can give you the list of what we have. Also in the high content core, we have screening libraries. So both single or small interfering RNAs and compound libraries. Many, as you can see, various libraries available to look at different things. And we have a liquid handling machine that can do all the movements for you. So basically you just bring us the cells and we can set it all up with whichever compound you want to use. Again, Mike is your best contact for this. And with that, I will say thank you. Hopefully that wasn't too much crazy information to dump all at once. And I look forward to answering some questions because that's really the fun part of this.